All right, so there were some questions on the Talk to Me forum and via email about problem 10 on quiz number 2, and then also a couple questions on problems from chapter 4 in your book. So the problem number 10 from quiz number 2, so this is asking you which biochemical method is most readily used to separate cytosolic proteins from nuclear proteins. So that the answer to that is differential centrifugation. So I have a whole slide on this and I talk about it in the lecture. So this is how you can separate different cellular fractions away from each other. So like in this figure right here, which I talk about in the slide, you basically lyse the cells open and you make a homogenate of the entire cells. You can then centrifuge them, and then heavier, more dense material is going to go to the bottom of that centrifuge. So that allows you to split up these fractions. So right here, we would have the nuclear fraction. So this is all the proteins and lipids and everything that were in the nucleus. So those weren't lysed when the outer cell was lysed. And you can even, centrifuge for more and get a mitochondrial fracture, or even more and get a microsomal fraction. And then the cytosolic proteins are going to be in, the solute basically in here. So the answer is not immunoprecipitation, because immunoprecipitation is only used to isolate one specific protein. So there's a lot of different proteins in the nuclear fraction, or in the nucleus in general, in a cell. So if you use differential centrifugation, you can separate cytosolic proteins from nuclear proteins. But if you wanted to only isolate a specific nuclear protein or a specific cytosolic protein, you could use something like immunoprecipitation. So immunoprecipitation uses antibodies that are designed specifically to bind one specific protein. So it could be one of many nuclear fraction proteins or one of many cytosolic proteins. But that antibody is only going to bind that protein. So you can basically incubate the antibody with your protein sample. It's going to bind to that antibody. You wash all the other proteins away, and then you isolate that antibody and release the protein from that antibody, and then you've isolated your protein. So differential centrifugation would be used as a very crude method of purifying proteins. So for instance, cytosolic proteins away from nuclear proteins or mitochondrial proteins. But immunoprecipitation is only useful for one individual protein. So take some time, maybe go back and look over those slides if you're still unclear about that, or just let me know. Okay, so there was also some questions about chapter 4 problems. So one of those was problem 6. And it says, name those components. So examine the segment of a protein, which is this one shown below. So A, what 3 amino acids are present? So we just look at the side chains. So that's going to be right here. So this is the carbon bound to the amino group and the carboxylic acid group on the amino acid. And then there's the side chain. So this is just a methyl group. So this is alanine, and its 1-letter code is A. So what's next? So now we actually have this peptide bond here, but I'm getting ahead of myself a little bit. And then we have the amino group and then another side chain, which is just a hydrogen. So that is the simplest amino acid, which is glycine, GLY and G. Then we have that carbon binding to the carboxylic acid group. Then we have the peptide bond. Then that amino group is binding to another alpha carbon. And so it has this side chain here. So this is one methyl group and one hydroxyl group. And so that is the polar amino acid serine. So SER and S. Okay, so alanine, glycine, serine. So of the 3, which is nearest to the N-terminal amino acid? So remember that we always write from N-terminus to C-terminus. And so this is the amino terminus, and so this alanine would be the amino acid that's nearest to this N-terminus of the protein. Number C, identify the peptide bonds. So these are going to be between those carboxyl groups and the amino groups on those proteins. So we have the amino group here. And actually, let's just do D as well. So where are the peptide bonds and where are the alpha carbons? So we have, this is actually a peptide bond here, but I'm going to put lines above these so you can see. So, because I already circled them. So this right here is the alpha carbon. It's bound to an amino group and a carboxylic acid group. And then that carboxylic acid group is bound to the next amino group. So this is the peptide bond. Okay, and so that amino group is linked to another alpha carbon. That's where the R chains are coming off. And so here would be the next peptide bond. And so that nitrogen is then attached to the alpha carbon. It's attached to a carboxylic acid. So this would actually be a peptide bond. And this would be a peptide bond here. So those are where the peptide bonds are and the alpha carbons. So the alpha carbons are going to be connected to the 4 different groups, except for with glycine. Okay, next one is problem 14. So this one says sweet tooth, but calorie conscious. So aspartame, which is the chemical in NutraSweet, so it's an artificial sweetener, is a dipeptide composed of aspartate and phenylalanine, in which the carboxyl terminus is modified by the attachment of a methyl group. So draw the structure of aspartame at pH 7. So let's draw the structure of aspartame in its fully protonated form. So it says that it's aspartate and phenylalanine. Well, let's start with the NH3 group. And so that's going to be protonated and carry a positive charge. So then we have a carbon. I guess I could draw the hydrogen. And then we have aspartate. So this is C, 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 O, O, O negative. But actually at this high of a pH, it would be protonated, so it would be OH. And then, so now we have the carboxyl group, and then we're going to start an H. So that's the peptide bond right here. And then we're going to get phenylalanine. So that's going to be C, CH2. And then we're going to draw the benzyl ring. Okay, so this C is attached to a hydrogen. And then we have C. So I already know, so the carboxyl terminus is actually modified by the attachment of a methyl group. So what that means is there's going to be a C double bonded O, and then O, and then CH3. So this is the fully protonated form of aspartame. And so it has a couple it only has 2 ionizable groups. So this is an ionizable group right here, and this is an ionizable group right here. So the pKa of this group right here is about 4. About 4. And then the pKa of this group is going to equal about 8, something like that. So it's asking us to draw the structure of aspartame at pH 7. So because pH 7 is above the pKa of 4 for this carboxylic acid group here, you would basically lose that hydrogen and then have a negative charge right there. So this would be the structure of aspartame at pH 7. So this group over here is not ionizable because it's been modified by that methyl group. So, okay, I hope those answered your questions. If they didn't, please let me know and I'll explain it differently. So thank you. Keep the questions coming.